Well, hey, AP Bio, this is going to be continuing on our discussion in the natural selection topic of evolution. And this is going to be all about the Hardy-Weinberg equation. Okay, so let's walk through the learning objectives together. So describe the conditions under which allele and genotype frequencies will change in population. Describe the conditions that will change, right? Again, evolution is the change in the genetic makeup. And so Hardy-Weinberg is a model for describing and predicting, right, good science, allele frequencies in a non-evolving population. So this population is non-evolving. Conditions, there's 5 conditions that we'll talk about in class, and these conditions are seldom met, but they provide a very valuable null hypothesis, right? So we can reject it, like, oh no, this population is not in Hardy-Weinberg equilibrium. We suggest that it is evolving. And so here's the equations for that. We'll practice through this. This is math kind of stuff related. And explain the impacts on the populations if any of the conditions are not met. For example, changes in allele frequencies provide evidence for the occurrence of natural selection. Again, evidence, you'll talk a lot about justification. You gotta use evidence. And small populations are more susceptible to random environmental impact than large populations. You know, and that relates to the bottleneck effect and all that we've kind of talked about before. So the Hardy-Weinberg equation is really kind of fascinating in the sense that it's providing an opportunity for us to actually measure evolution. So genetic variation is required, right? We get variation through mutations to evolve, but it does not guarantee it will. One or more factors that cause evolution must be at work. So a population, again, knowing what a population is a group of individuals that live in the same area and that interbreed and produce fertile offspring, okay? They might not be geographically isolated, but individuals still tend to breed with members of their own population. So for example, these 2 caribou populations are not totally isolated. Still, members of either are only most likely to breed within their own populations. And so the frequency of an allele in a population can be calculated. For diploid organisms, the total alleles at a locus is the total number of individuals times 2, right? Because you get 2 copies of each gene that you have, 1 copy from mom and 1 copy from dad. And so homozygous dominant, like let's just write them out, right? Homozygous dominant, heterozygous, homozygous recessive. And you can see that in a population, homozygous would donate 2 capitals, heterozygous would donate capital and a lowercase and 2 lowercases to the gene pool. And so the gene pool is all the alleles of all locations in a population, of all genetic locations in a population. So an allele for a particular locus is fixed if all individuals in a population are homozygous, which makes sense. How can you pick anything else if all you have is homozygous? And so, for example, and this imported incorrectly, so let me look at my computer here. But P plus Q equals 1. So this is P plus Q equals 1. So P and Q are used to represent their frequencies. So the frequency of all alleles in a population add up to 1, okay? So for example, we're going to walk you through an example here. So for example, consider a population of wildflowers that is incompletely dominant, which means there's a blending for color. You have, this encodes for red, there's a blending here, and this encodes for white. And so the number of each allele, well, 320 times 2 is where they get that. They got that from this one, right? And so 320 times 2 plus 160, why is it 160? Because these individuals have 160 red and alleles and 160 white alleles. So that's 800 and 200. And then this is showing you like the different flowers. And so the frequency of that is P is 0.8 and Q is 0.2. And they just figured that out from doing the math, right? Simple math here. And so the Hardy-Weinberg equation describes the genetic makeup expected for a population as not evolving. And you're like, well, you know, if the data observed for the population differs, then the population may be evolving, right? You do more statistical analysis. And so a population in which mating is random and none of the mechanisms of evolution are acting is in Hardy-Weinberg equilibrium. These allele frequencies remain constant. But if allele frequencies do not remain constant, then you could assume or, you know, suggest that evolution may be occurring. And so 0.8, 0.2. And so you can see right here, all these alleles, assuming man each time 2 gametes come together, there's an 8% chance that an egg will carry a red allele and a 20% a white, and same for that, 80%, 20%, okay? And so you can figure out the frequencies of genotypes can be calculated using the rule of multiplication. So 0.8 times 0.8 is 0.64. So 64% we would get red. 32% we would get pink. 4% we would get white. Okay? And this is what it looks like. From a Punnett square, you can see, 0.8 times 0.8, 64% right here. 16 and 16, you add those up, 32% and 4%, and of course that all adds up to 1. And so evolution can be measured, number of alleles in a population divided by the total number of copies of all alleles in the population, okay? And so P is the frequency of A, so dominant, or the capital, it doesn't necessarily have to be dominant, and frequency of the recessive or the other possible, okay? And so P plus Q equals 1. And this is known as genetic structure, the frequency of different alleles and genotypes in a population. Again, we're looking to see if populations are evolving or not. So the Hardy-Weinberg equilibrium is a model in which allele frequencies do not change. Again, see that word not change. So we're not changing anything. And so there has to be 5 assumption of this Hardy-Weinberg equilibrium. Here's a little amoeba sisters picture, right? No selection, everybody has an equal chance of mating. No mutation, which of course never happens. No migration, large population, and random mating. Everyone has an equal chance. Okay? So this is just reworded, but no mutations. So gene pools, modified mutations happen. So you're like, well, this is crazy. Well, again, this is a model, right? Is enough happening for evolution to occur? You're like, these conditions are always never met. That's true, but we have to have, and then we'll do statistical analysis to figure it out. Is it in equilibrium or not? Random mating, no natural selection, which means everybody has an equal chance of surviving and reproducing. Extremely large population size, so we're talking crazy large. Small populations, genetic drift can affect small populations. That's like bottleneck effect, founder effect. We talked about those already. And then no gene flow. We're not moving into or out of populations at all. No immigration or emigration. So because of one of these influences are typically present in real population, the Hardy-Weinberg principle describes an ideal condition that against which the effects of these influences can be analyzed. And so you can see right here that homozygous dominant is p squared, heterozygous is 2pq, the frequency, and homozygous recessive is q squared. And so this is the formula. P squared plus 2pq plus q squared equals 1. And remember, for alleles, P plus Q equals 1. So those are the 2 formulas that you have to know about. And so we're going to practice this a lot in class, doing practice problems, like I said. We're doing notes at home, practice problems in class. But we'll go through some practice problems here as well. So in real populations, allele frequencies and genotype frequencies do change over time. And evolution is occurring, usually, but is enough change present that we can say, like, okay, well, yes, this is for sure happening. All right, so let's do a check here. The first one, a population of octio plants has 4 alleles at the dopa locus with allelic frequencies of, here's all the allelic frequencies, okay. And the genotype frequencies roughly consistent with the Hardy-Weinberg equilibrium. This is a description of what? Well, I never said this word, okay. It's not fitness. Debatable, right? But we talked about it, genetic structure, okay. Let's do a quick check for juniper trees. 3 different alleles, C1, C2, and C3. If the frequency of C1 is 0.3, well, they all had to add up to 1, right? And so go ahead and try to solve these before I solve them. So go and pause. The frequency of C2 cannot be greater than 7 because there's 3 alleles, right? If it's greater than 7, we're already above 1, and we haven't even counted another allele yet. The FY3 gene plays a role in immune responses in flies. In one population, there's 2 alleles. The frequency of one allele is 0.6. What is the frequency of heterozygotes? Well, here's where we get our equations. P plus Q equals 1, and then P squared plus 2PQ plus Q squared equals 1. Let's just say P equals 0.6. Q would have to equal 0.4. How do I get that? 1 minus 0.6 is 0.4. And so heterozygotes, though, which one is heterozygotes up here? Well, this is homozygous dominant. This is homozygous recessive, so this is heterozygotes, right? So it's important with these problems, you really have to know what variable are you given and what are you looking for and what are you solving for. So we're solving for 2PQ. So 2PQ is heterozygotes. Then we just plug in the numbers. 2 times 0.6 times 0.4, well that's 0.24 times 2, and that's 0.48. That's the answer there, okay? Several populations of mustard plants are sampled for variation at the F locus. The percentage of each genotype for populations AB are summarized in the table above. Which population shows a heterozygote deficiency? So what we expect a heterozygote to be, which one is showing like, man, there's some sort of fitness level that's lower for heterozygotes for some reason. It's lower than what we expect on paper, okay? And so you would have to work this out, and since we're doing it, let's go ahead and do it together. So whenever they give you this, you always solve for Q squared, okay? You always try to start with recessives, okay? Because when they give you dominant phenotypes and all that, but just for the record, let's just start with Q squared, so Q squared equals 0.2 for population A. To solve for Q, you would square root both sides. Let's pull out my calculator here. Not ready for that. And well, actually, let me pull out this calculator. Sorry, I'm just talking myself through what I'm doing that you can't see, which is always annoying. And all right, so I have to charge battery. Okay, and so so the square root 0.2, 0.45. 0.45. P would have to be 0.55 because P plus Q equals 1. And then you just times these numbers together and then times that by 2, right? So 0.45 times 0.55 times 2 is 0.50. 0.50. 60% is greater than 50%, so it's not population A. All right, let's do population B now. Ooh, need a bigger eraser. Population B, Q squared equals 3%, or 0.03. Square root both sides. Square root of 0.03, Q equals 0.17. P equals 1 minus 0.17, 0.83. And then you just do 2PQ, right? 2 times 0.83 times 0.17, and that's 0.2822, whatever, so 28%. 28%, 34 is higher than 28, and so that is also not population B. And so it has to be population C that is deficient, but let me just show you the math real quick. Q squared equals 0.35, square root both sides. Square root of 0.35, square root point, my bad, 0.35. 0.59. P is 0.41. You times those numbers together and times it by 2. 0.59 times 2 times 0.41. Our heterozygote should be around 48%. It's not, it's at 30. So the answer is population C. Okay. Next practice problem. Again, practicing all this out would be good. You discover a population of guppies and find it to be at Hardy-Weinberg equilibrium. So it's meeting the 5 assumptions of the Hardy-Weinberg equilibrium. At the S locus, a specific gene. Suppose there are 2 alleles at this locus and the frequency of one of those alleles is 0.4. Let's just pretend like that's P. P is 0.4, Q is 0.6. Why? P plus Q equals 1. Of the individuals that carry at least one lowercase allele, what fraction are homozygotes? Okay. Well, it's asking you, so it's asking you what fraction, and then, yep, I made a mistake. Whoopsie doopsies. I would go back and record this, re-record this if I was on a computer, but I'm not, I'm recording this on an iPad. So the lowercase allele is 0.4. So hey, lowercase equals Q. So easy to get these wrong. That's why you got to read. And so Q equals 0.4. So P equals 0.6. That's crucially important. We're going to have gotten it wrong if we did that. And so, yeah, so let's figure out P squared, 2PQ, Q squared. Okay? Q, 0.4 squared, 0.16. 0.4 times 0.6 times 2. 4 times 0.6 times 2. 0.48. And then 0.6 squared is, 0.36. Okay. So it's asking you about these two, right? So, because remember, this is standing for capital S, capital S. Let me do different colors. This is standing for the genotype capital S, capital S. This is capital S, little s, and this is little s, little s. All right, so it's asking you, of the individuals carrying at least one S allele, so the total of these two, right? And so what is that, 0.64? 0.64 is the total. What fraction is 0.16 over that? And that is 1/4. Okay, 1/4 would be that. And so go ahead and go through these concept check questions. And also, we'll go through those answers. And then, yeah, definitely do the science practice as well, because we'll go through these as well in class. All right? Work hard. Do your best. Much love in Christ, guys. Proud of you.